So this is our first video on passive transport. This goes along with the cell transport guided notes packet that you should have in front of you while you watch this. This video is going to start out on the second page. About halfway down, you'll find a section called Part 2, Transport Across the Membrane. It's bolded and underlined. We're going to fill in the notes for that for passive transport. There are two types of transport or movement across the cell membrane. There's passive, which does not require energy, so no ATP is required. And there's active, which does require ATP. It does require energy to move things across the membrane. The three types that we are going to be talking about are diffusion, osmosis, and facilitated diffusion. Our first type, diffusion. This is the movement of small, nonpolar molecules across a semi-permeable membrane. So there's kind of our textbook definition of what diffusion is. So on the guided notes, anything that you need to record is bolded and in red. If you need to pause the video in order to catch up and watch them, please feel free to do that. So small molecules and nonpolar molecules. Nonpolar molecules meaning they have no regions of charge, so no charged areas on them, meaning that they're going to be able to kind of slip right through the phospholipid membrane. They're not going to interact with any of the charged parts. Because diffusion is passive transport, here we have our criteria for everything that's passive transport. The molecules are going to move from high concentration to low concentration. So in an area where there's a lot of them, they're going to spread out, move to areas where there's less of them, and it's not going to use ATP, so no energy required. And this is going to continue, so this movement of molecules from areas where they're crowded, so high concentration, to areas where they're more spread out, low concentration, until we reach what's known as an equilibrium. Equilibrium is when you have the same amount of a substance on both sides of the membrane. Now, there are lots of different types of equilibriums. Equilibrium basically means a stable condition where everything is equal. So if we look at equilibrium, the word itself, the first part, equal, kind of tells us what's going on. So in the diagram over here on the side, we notice that we have our phospholipid bilayer. So we have our cell membrane that runs through the middle. On one side, we have a lot of glucose molecules. So this would be an example. So I'm looking at the far left-hand side. This would be an example of something that is not in equilibrium or is in what's called disequilibrium, because we have a high concentration on one side and we have none or very, very low concentration on the other. So in equilibrium, these molecules are going to pass straight through your cell membrane until we eventually get to a point, so our picture on the far right, where we have roughly the same number on each side. Now, in equilibrium, that does not mean that everything stops moving. So that doesn't mean that the glucose that are on one side stay on that side forever and ever. What it means is that we have what's called no net or total movement, meaning they still move back and forth across the membrane, but about an equal amount are going in as they're leaving. So there's no total change in any direction. So again, diffusion. These small nonpolar molecules are going to occur directly through the phospholipid bilayer. And any diffusion where a molecule is literally small enough that it just can squeeze right through the phospholipids to get inside the cell is referred to as simple diffusion. So two examples of molecules that do this simple diffusion are carbon dioxide and oxygen. So if we remember talking about the respiratory system, we talked about how oxygen diffuses into the blood. So it just squeezes right through the phospholipid bilayer into the blood. And we talk about carbon dioxide diffusing into the lungs. So same thing, squeezes through those phospholipids to get into the lungs. Now, water is so special that it actually has its own type of transport. And this type of transport is called osmosis. So osmosis is the movement of water across a semi-permeable membrane. So it's basically the diffusion of water. So our same rules are going to apply here, meaning since it's passive transport, it's diffusion, it requires no energy, and water is going to move from areas of high concentration to low concentration until an equilibrium is reached. So in the diagrams on the bottom, imagine that we have a higher concentration of water on the outside, labeled A at the diagram, and we have less on the inside. So water is going to diffuse or move through the membrane into the cell until eventually we reach an equilibrium where there's roughly the same amount on each side. And again, this doesn't mean everybody stops, nobody moves anymore. It just means there's no net movement. So as many go in as come out. Term alert. So one of the things you're going to come across a lot in this unit is a term called concentration gradient. We know that concentration means the amount of stuff dissolved into a solution. And the term that we have for that stuff is solute. So when I think of concentration, I always think of making like those sugar drinks from like a powder. So, if you have a jug of water and you wanted to make something with a low concentration, then you just put a tiny little bit of your mix into the water and it kind of has the faint taste of it. So that's a low concentration. There's just a little bit of powdered mix for the amount of water you have. If you wanted to make a high concentration, you would put a whole ton of that mix into your water and mix it up. It's going to taste a lot stronger because the amount of mix, which is your solute, has a higher concentration. There's more of it to the amount of water that you have. When we talk about things moving from areas of high concentration to low concentration, you'll hear this term called moving down the concentration gradient. And it basically means that things are moving from an area where there's a lot of them, so they're concentrated, to an area where there's less of them, so less concentration. So when a concentration gradient exists, water is going to move in and out of the cell to attempt to reach equilibrium. So we have 3 terms for describing the movement of water across concentration gradients. So where you should be right now is on the back of your notes, you have a chart that has, and if you look in the far left-hand column, you'll notice it says isotonic right at the top. What you are going to do is this diagram that I have here over on the left-hand side, you're going to sketch this inside that first box that's on the top. Features that you need to pay attention to is you need to draw your cell membrane, which is this thick white line that runs through the middle. And what you're doing to depict an isosonic solution is you're showing the same amount of solute, which is green circles, on the inside of your cell as there are on the outside of the cell. Because isotonic means that we have solutions of the same strength inside and outside the cell. So because we have same amount outside as we do inside, we get that no net or total movement of solute in or out of the cell. So we still have maybe one goes here, maybe one comes here, but it's equal. So the amount of movement out equals the amount of movement in. So that's called isotonic. The next one we have is hypertonic. So hypertonic is when we have a greater strength outside the cell. So here, again, you're going to need to sketch this. Please pause the videos when you're going to sketch. Don't keep sort of running it because you'll end up behind. So here's your cell membrane. Here's your solute being shown in your green circles right here. So this is the stuff dissolved in the solution. We notice there's less of it inside the cell than outside the cell, and that makes this a hypertonic solution. I always think of this like when you're hyper, it means you have too much energy. So when the solution is hypertonic, it means it has too much stuff or too much solute dissolved in it. Now, in this case, water is going to move out of the cell to try and make equilibrium. So it's going to try and move out of the cell to spread out the amount of solute, to dilute the amount of solute in here. Now, when that happens, so when water moves out of our cell, the cell is going to shrink because it's got less water inside the cell. Our last one is called a hypotonic solution. So in a hypotonic solution, this is the exact opposite of your hypertonic. So in this instance, the cell has more solute. It's got more stuff dissolved in its cytoplasm than the solution outside of the cell. What happens here is that, again, water is always going to move to dilute. So water moves in the direction of greater solute concentration. So in this case, we have more solute inside our cell, so water is going to move into the cell. This means that our cell is going to get bigger and bigger. If the solution outside here is hypotonic enough that it causes enough water to go inside the cell, what can actually happen is the cell can burst. Now, this doesn't happen 100% of the time. It only happens when a cell is in an environment where it's in a very hypotonic solution. So it is a lot more concentrated on the inside of the cell than it is on the outside of the cell. A lot of water rushes into that cell and can actually pop it. And we call that popping of a cell, we call that lysing, L-Y-S-E.